Our next topic of discussion is the different stages of drug development. Drug development is not an easy one. It takes a lot of time and we have to spend a lot of money for the development of each drug. This lecture is meant for giving you information about what are the different stages of drug development. Drug development involves a lot of actually stages. You can see that here before as a beginning of drug development, to make one approved drug, you can see that it start with more than 20,000 compounds. Maybe sometimes we have to take around 1 lakh compounds. Okay, so it start with 1 lakh compounds. Then we can call that this is called basic research. Okay, after doing some basic research, what we do is that we had to screen our targets to 5,000 to 20,000 molecules. This is actually there we are going to the next stage, that is called drug discovery stage. In the drug discovery stage, we will be getting around 250 molecules to the preclinical stage. And among 250 molecules, after studying the pharmacodynamics and kinetics, we select only 5 molecules to next stage, the clinical stage or clinical research stage. And of the 5 clinical drugs used in the clinical trials, only 1 is getting approved. These are the basic things. So for getting 1 approved drug, we can see that we have to start with anyway about 50,000 to 1 lakh molecules. And these are the timeline of the different stages of drug design. The basic research takes place 2 to 3 years. And the basic research deals with the discovery and origination of the new compounds that become drug candidate. That becomes a drug candidate. That's one of the important things that we do in basic research. And here what we synthesize new compounds, and we explore different target molecules, and we screen the molecules for the desired biological activity, and we do some of the formulation research also. The next stage after basic research is non-clinical studies. It will also span around 3 to 5 years. This stage is actually used to study the new efficiency of the new compound. Okay, so here we do the pharmacological efficiency studies. Okay, and we study the pharmacokinetics. Pharmacokinetics means what the body does on the drug, what the drug does on the body, and actually then we do the pharmacotoxicity studies also. This is very important for the pharmacological formulation design, developmental research and further development, and the study method development, and also for the investigational drug quality assurance. Okay, this is called the non-clinical studies, and these studies actually is done in animal models, basically, by in vitro and in vivo methods. After that, now it's around 3 to 5 basic research took 3 years, and non-clinical studies took around 5 years, total 8 years. And after 8 years, we go to the clinical phase. Clinical phase span around 3 to 7 years. And you can see that here there are 3 types of, actually there are 4 types basically, but there are for a common point of view, we need to have only 3 phases of the clinical trials. After the clinical trials, we'll discuss in detail later. After the clinical trials, the new drug application is submitted to the agencies, and it takes around 1 year to complete the analysis because they demand a lot of data. And after that, the approval of the new drug is sanctioned. And after the approval of the drug, people don't sit idle, and they will constantly search for any problem and possible side effects and long-term side effects. And that's called post-marketing surveillances and clinical studies. This actually comprises of the phase 4 of the clinical trials. So there are 4, 5, 6 different stages actually in a drug discovery. The first stage is actually what we call that to be the target selection stage. And the second stage is the lead discovery stage. And third stage is the medicinal chemistry stage. Fourth stage is in vitro studies. Fifth stage is in vivo studies. And sixth stage is the clinical trials and therapeutics. The first 2 or 3 stages comprises the drug development process. And the first important phase of the drug development process is the target selection. Then the lead discovery, medicinal chemistry, in vivo studies by using the bacterial cultures or cell lines, then in vivo studies by using animal models, then we go to the clinical trials. And actually, this in vivo studies is very important, and this is important for model development, target validation, and measure of the target effects, and also for data management. The first stage of the process is actually, in fact, the target selection. Target selection in drug discovery is actually the decision to focus on finding an agent with a particular biological action that is anticipated or expected to have a therapeutic utility is influenced by a complex of many features. Okay, the first one is called target identification. Actually, here we identify the molecular targets that are involved in the disease progression. For example, in the case of some cancer cells may produce some proteins. Okay, and those proteins may be some target for our attack. If there is no such protein, the progress of the cancer can be prevented. So, the first thing we have to find is that to find target where the drug is going to act. That is called target. For example, some anti-cancer drugs attacks the DNA of the cancer cells. Okay, for example, cisplatin, it attacks the DNA of the cancer cells. And actually, the first very important thing is that to identify the target. Second one is actually target validation. Okay, so a target validation is actually to prove that manipulating the target can give the therapeutic benefit to patients. Okay, after creating a target, we have got the drug. We test the drug with the target, and we have to find that even the molecular target, when the drug candidate comes and meets with the molecular target, it could provide some therapeutic benefits. Some of the important targets are G protein coupled receptors. These are some type of protein receptors in our body. Most of the 45% of the drugs targets on that. Enzymes around 28%. Hormones and factors. For example, in the case of breast cancer, there are some special type of hormones produced in the breast, and some of the drugs actually they attacks the effectiveness of the hormone. And next was ion channels, and also the nuclear receptors also play a very important role. The second stage is the lead discovery stage. At the lead discovery stage, there are two things that you have to consider here. The one is the lead identification and the lead optimization. In the lead identification, around, as I told you before, around 5 to 5,000. It's not actually 50,000. 5 to actually 1 lakh molecules, especially only in presence of COVID and a very emergency time only. We usually study around the small number of targets. But in other ways, you can see that we take around 50,000 to 1 lakh compounds in the laboratory. And among them, we pass only a very few to the next stage. Okay, so once the therapeutic target has been identified, scientists must find one or more leads. That means some of the molecules that can interact exactly with the target. Okay, and multiple targets are also sometimes selected to get better results. And first of all, biologics ensure that the chosen compounds have desired therapeutic or antiviral effects on the target. They test the compounds for the toxicity. And in the case of the vaccine, the viral activity is also measured by using in vitro, cellular, or tissue models. Finally, they check the bioavailability or in vivo animals, in vitro and cellular and unknown tissues. And these data are very important for the drug licensing. The second one is lead optimization. Lead optimization takes place around 4 to 6 months. From the lead identification, we have selected some compounds, and chosen compounds are examined in the laboratory in order to perfect the physiological properties. And we study the pharmacokinetic behavior of the compounds, and among them 20 molecules are selected and tested further on. This purpose of the stage is to optimize the molecule or the compounds that demonstrate the potential to be transferred to the drug. and retaining a very small number of molecules in exchange, because the next stage studies involves a lot of time and money, and because of that, to prevent the unnecessary burden on the patients or the price of the drug, the pharmacy, pharmacology, the medicinal chemist minimize the number of charges that to be given to the next stage. As there are a lot of information required for the filing application for the drug, the scientists use very important techniques like spectroscopic methods, X-ray crystallographic methods, in silico computational modeling method, that means molecular docking and dynamics method, and also they study the selected molecules link themselves with the therapeutic target, like the protein or an enzyme. This data is very much helpful to the medicinal chemist to modify the structure of the selected molecules or compounds. Okay, these are the important things that the people do. And the third step is the medicinal chemistry step. Now, the medicinal chemists prepare or select appropriate molecules for biological evaluation. And it is found active, they could serve as the lead compounds. They then evaluate the structure-activity relationship. That means they select the molecule and modify the molecule to make analogous molecule, and they study the different activity and safety of those molecules by using structure and activity relationship. Now, medicinal chemistry is not actually a chemistry field; it's actually interdisciplinary field, and must therefore a medicinal chemist should not only understand organic chemistry, but also they should undergo some process development, some biological activity for pharmaceutical activity, etc., to go further. Earlier, actually, people used to get data only from in vivo testing. But now we can see that a lot of in vitro screening, combinatorial data, and other things are important, and they are used actually for the evaluation of the medicinal activity. Although these new technologies presents many opportunities, there are a lot of safety requirements have been raised these days, and these safety requirements have been is present as in some unanticipated hurdles for the transformation of the molecule to the drug. Next one is the in vitro studies. In vitro studies are essential part of the research geared towards the discovery of the drug candidate. Actually, these are actually used not in animal models, but in other models. For example, in the case of antibacterial studies, they don't put the bacteria in the animal model, but instead they culture the bacteria in a petri dish and we use the drug. In the anti-cancer drug discovery, there are cell lines available for each cancer, and the drug is used against each cancer to get to check whether it is having the effectiveness. Diabetes also, they study with the different markers that's already available. And also the in vitro studies sometimes actually include the study of absorption, how the drug is absorbed, and the metabolic stability of the drug, elucidation of the elimination pathways that depends upon the type, etc. Okay, and also some of the ability of how the drug interacts with some of the very important enzymes in the body, like CYP, cytochrome oxidase enzymes, CYP 450 enzymes, and also the metabolite profiling in various model species and humans. Okay, these are not necessarily conducted following regulatory statute, but in case these studies are essential for the submission of the initial drug, new drug report or IND report for both the preclinical and clinical stage. In vivo studies, actually, it involves that it is used to confirm the information that we get from the in vitro studies. This is done in a living organism. In clinical trials, this happens within animal models. Clinical trials in vivo studies can be used in either animals or in humans. Okay, and it's actually it's confirmed in parallel with humans and other animals, especially dogs, pigs, etc. And also our ancestor, supposed to be ancestor monkeys and rhesus monkeys. In vivo studies are able to address the major limitation of in vitro studies. They are able to demonstrate the impact of the pharmaceutical substance on the body as a whole, rather than how it's impacted on the isolated cells. This allows in vitro studies to better visualize the potential interactions, which can prove its predictions for safety, toxicity, and efficiency. And in vitro studies are having some drawbacks also. Actually, they also have to make a major setback. In vitro studies with significant ethical concerns, particularly for preclinical studies. We are using animal models, and animal models. People sometimes indiscriminately use animal models. There are a lot of what we can say is that pro-animal actually activists, and nowadays for doing any research by using animals, you have to have with the backup of the ethical committee. Every institution doing this type of research should have an ethical committee where the animal department, veterinary department people, even collector is also present in the meeting. Like in vitro studies are also undergoing a large modification, transformation. And emerging technologies are actually, we make actually new animal models to conduct, we genetically modify even mice. Okay, and we make some type of mice such that the disease is present from the birth itself, and that reduces the time span of the research, and also it makes the research cheaper, because we need not require to use that the high amount of animals, a large number of animals here. Next one is clinical stages. Clinical stages actually, it involves actually 4 phases basically, and the first phase is limited to actually 20 to 80 participants, and the next stage is 200 to 300 participants. Phase 3, that's the most important phase, is limited to 1,000 to 3,000 participants. After that, normally people give the final report to the FDA. FDA means Federal Drug Agency in America. In India, there is Drug Controller General of India, and we file that to that people, and they confirm the safety and effectiveness of the drug and allow for marketing. Even though, as I told you before, even though we subject the we allow for the marketing, it doesn't mean that everything is over. For at least 5 to 6 years, the post-market surveillance is also done by taking data from the patients. That is called phase 4, and around 1,000 people participate in the phase 4 trials. And even though in the first stage, preclinical stage, we have seen the in vitro and in vivo studies here, and to enter to the phase 1, we need permission. Okay, so if we had to file a IND here, initial new drug application we had to file here. We had to give all the data from the animal models and vitro studies to the regulatory agency. As I told you, for America it's FDA, but for India it's Director General of Drugs, Drug Control and General of India. And it enters then only it enters the first phase. Even in the in this stage itself, they will make a rigorous check of the different properties of the molecule. Then only it enters the different stages. That's why the drugs are very costly. It takes about 15 to 16 years to get a good drug to the market. So the companies are spending a lot of money on that, and they will take the money back from us, the customers. The clinical trials are actually used as a prospective biochemical or a behavioral research study of human subject, which is designed to answer specific questions about the biomedical or behavioral interactions. For example, vaccines, drugs, treatment, the devices used for the treatment, and actually new ways of using the non-drugs. That is called the repurposing of the new drugs and treatments and devices. Clinical trials are used to, for example, the repurposing of the drug. During the COVID time, scientists first went through a large number of the non-drugs. They screened the non-drugs against the coronavirus, and that's why remdesivir, ivermectin, and albendazole, etc., first recommended for the COVID treatment against the COVID. Clinical trials include behavioral human subjects research involving an interaction to modify the behavior. For example, in the case of antipsychotic drug, they study the behavior of the compound. Okay, so the human subjects research to develop, use human subjects to develop or evaluate the clinical laboratory tests. Like imaging or molecular diagnostics may be considered to be clinical trial. The test will be used for medical decisions. So there are different stages. The first stage is phase 1 clinical trial is generally done in the healthy individuals to study how the drug is interacting with the people in the human body. It takes around 20 to 80 people. Normally, it is done in healthy people. It is not for therapeutic purpose. It is only for getting the pharmacodynamic data from the people. Okay, it is alleged that in some countries like Latin America and America, these studies are done in jail, prisons, and and the people who participate in such studies are exempted or given relaxation of a few years for the prison sentences. It is not sure, it's not a public document. I just want to tell you only. And second stage is phase 2 stage. And phase 2 clinical stage studies the biomedical and behavioral interaction with a large number of people. Maybe around 700 to several hundred people have been tested in this stage. Here we deal with patients, a small number of patients. Okay, and this phase is not done only in a single country, and maybe it is spread over different countries, different places, and like that. And the phase 3 studies the actual efficiency of the study or the behavioral intervention to the human subjects. And that involves a good number of people. It may involve around 2,000 to 2,000 people, and it is spanning in a large number of nations. For example, sometimes in India, North India, South India, in Russia, in Europe, in Africa, America, Latin America, in all places, these clinical stages are conducted for a single drug. And there are some hospitals in India itself who regularly participate in the clinical trials, because we can't say that why they are not doing like that, because it's actually a part of the drug development process. They are participating. For example, in our near Ahmedabad hospitals participates in the different clinical trials, and also in some of the medical colleges, and also very famously CMC Vellore. That's why they have got very good curing rate. It's very famous. CMC Vellore participates in a large number of clinical trials. And you can see that intervention is found to be very crucial and very important, and most powerfully productive also. And after doing the phase 3 trials, the studies are actually reported to the regulatory agency, and the regulatory agency checks the every data very carefully and meticulously, and after they approve the drugs. Okay, and then after that, this is allowed to market. At the phase 4, after being marketed, again there will be side effects, a long-term side effects, and this is actually monitored regularly, and this is actually given back to the pharmaceutical developer. These are the different stages of the clinical trials. So we are seeing the different stages of drug discovery, and you can see that these are the different stages. We are seeing the basic research, non-clinical studies, clinical trials, the application, approval and launch, and the post-market surveillances. These are the different steps. We take the minimum number of time. I can say the minimum number of time basic research 2, non-clinical 3, total 5 years, clinical 3, 8 years, and 1, 9 years. And if you take the minimum time, it will take around 9 years to launch a new drug. If you take the minimum time span, take around 8 to 9 years to launch a drug. Okay, so thank you very much.